First of all, this is my first video. I am not a native speaker and I will definitely make mistakes in the language. I apologize in advance for this, but I hope for your support if you like the video. According to surveys, about half of the people on the planet do not trust GMOs. Over the past couple of years, this figure has reached half in America. I wanted to look into this and tell you what GMOs really are. Is this our salvation or our curse? I want to go from small to big, from bacteria to plants, then to the animals, and in the end we will draw conclusions. But first, I will express one thought. Our universe is designed so that the time goes only forward. Therefore, matter is forced to move somewhere and collide with itself. It happens randomly and endlessly. And the trick is that matter can connect with each other. Atoms with atoms form molecules. Molecules sooner or later, as a result of endless and random selection to each other, combine into amino acids. They will then assemble into proteins, DNA, etc. Sooner or later, such a combination will be selected that will receive the property of self-reproduction, simply because the endless selection leads to inevitability of any event. Our life is made up of proteins or amino acids. These are the bricks of the building, but not to do it randomly every time, getting different mountains of bricks instead of walls. You need to write down a working plan and pass it on to your descendants. We need a text or a code, which is DNA. In the context of this topic, it is important to keep this in mind. But then it's more important. You should understand that a lot of interesting things happen to DNA. And I am not talking about mutations. I am talking about how cells are constantly working with DNA. I will tell you about different mechanisms so that you understand how it works approximately, and we will not return to this topic. With DNA text or code, something is permanently happening. Life never stands still. For example, you need to copy DNA to divide a cell in two. For this, there is a protein that cuts DNA in half. The molecule is double-stranded. Each chain already has a complete set of information. You disconnect these chains and give each half of the dividing cell. And then the new cells simply complete the second chain. And this protein for cutting DNA in half is a whole tool. It's like scissors which we can borrow from nature. Or another situation. There should be a certain amount of protein around the cell that it produces. More is meaningless, and if less, for example, the cell will die or something. How to regulate it? Exactly the same. There are proteins that turn on and off the reading of information from genes. Now protein assembly has stopped, and now it has continued. There are proteins that cut out genes, correct errors in mutations, etc. There are hundreds of situations and proteins, and we can take and use them for ourselves. But there are other mechanisms, viruses, that empty their DNA into our cells. So that cells by mistake create new copies of viruses. Scientists have come up with the idea to replace the text in these viruses so that our cells create not new copies of the virus, but the proteins we need. Bacteria, moreover, have additional DNA, it's called a plasmid. This is a way of exchanging information. One bacterium transfers a plasmid to another, like a flash drive, and that's it. Agree, it's quite difficult to insert information directly into the hard drive, trying to scribble something on it. But if you can replace the info with a flash drive and slip it to someone, it's very convenient. On the other hand, it's not necessary to take something from nature. You can do what people love the most. A gun. A gene gun. The essence is this. Small particles of heavy metals, such as gold, cover DNA and shoot at high speed at the cells we need. These mini projectiles break through everything in their path and get inside the cells, into the nucleus, into the mitochondria, everywhere. The method is somewhat random and not very accurate, but still working. You can literally slap the words you need into the text with a hard force. Somewhere it will fit the meaning, you will see these cells and continue to multiply them. I list all this to the fact that quite a lot of mechanisms are already known and scientists take and use them to edit DNA. So we have long been able to change the randomly created text to suit our needs. And every year there are more and more tools and they are getting better. The easiest way to work with bacteria. Almost all above methods can be used there. A good question is what to change there and why. Actually, bacteria are new factories. Once there was an industrial revolution, and this is, if you want, micro-industrial. And these factories can build anything. The classics are medicines. One of the first hormones found and studied was insulin. People quickly figure out that diabetes occurs without insulin, and that the solution to the diseases is generally simple. It is necessary to give someone else's insulin. Fortunately, we are all creatures very similar. So many animals have insulin. You can borrow it. The hormone is produced by a special organ, the pancreas. The only problem is that a lot of things stand out there. To get exactly insulin, it is necessary to destroy all the other cells of the pancreas. That's what scientists did with the dogs in the 20s. Just imagine, animals were opened. Half of the pancreas was destroyed, dooming to death, and they left a tube sticking out of the stomach, from where the insulin flowed out. And then there may be an allergy to foreign insulin, and then it stops working because it is a foreign protein to which our body develops immunity. As an experiment, okay, but as a full-fledged solution, not really. Fortunately, DNA is decrypted in the 50s. So people already understood how to work with it. And in the 70s they figured out how to inject a piece of DNA to bacteria, containing information about the structure of human insulin. That's why I told you that there are many options for how to inject the genes we need. In order not to stop and talk about them every time, here with the help of plasmids, and here a virus was used, and here a gene gun, and here the CRISPR-Cas, etc. So yeast or E. coli were given instructions on how to create human insulin, and these genetically modified bacteria have been creating insulin for 40 years. So that you understand the magnitude of the importance of this GMO current. Now there are 10 million people in the world who need insulin. Previously, when making such a diagnosis, a person died quite quickly. I am sure that the opponent of GMOs will change his opinion if it suddenly turns out that he has insulin-dependent diabetes. And there are quite a lot of such examples of medicines. For example, dwarfism. A common reason for this is that growth hormone is not produced in one part of the brain. The animal analog is almost not suitable. Therefore, in the 50s, when the hormone and the disease were just being investigated, the parents of such children in the USA had to negotiate with the pathologist so that he cut out this part of the brain, then with the biochemist so that he cleanses the drug, which should have entered more than once. Fortunately, then the government began to deal with this, but there was still not enough material from the corpses. Only the most severe cases were treated. It's so good that bacteria can synthesize our growth hormone, which they have been doing since the 80s. It is not necessarily the disease can be caused by the absence of hormone. This may be, for example, a specific anemia, for kidney diseases with dialysis, or chemotherapy, for example. The body begins to urgently replenish the lost red blood cells that carry oxygen. This is regulated again by the hormone erythropoietin. Well, in certain cases this hormone can be introduced from the outside to spur the cultivation of new red blood cells. And you can get the hormone again by inject the gene to the bacteria. By the way, more than that, erythropoietin is also used as a doping. Muscles need oxygen for energy. More red blood cells, more oxygen, more energy. So erythropoietin is considered doping. and one famous cyclist was stripped of all titles for such doping. I hope there are enough examples of medicines, but there are other things. Therefore, let me remind you what gas and fuel are. These are processed by tens, hundreds of millions of years and pressure the bodies of our ancestors, but mostly algae and plankton. And now imagine, we will definitely burn it all sometime. It is clear that we will switch to other energy sources, but in general oil is needed not only as a fuel. Oil has many other possible implementations. Oil is still needed, but let's say it's over. Or another argument, the extraction of fuel is quite expensive. Anyway, you already understand what I am leading to. Of course, natural oil is more difficult to obtain, but to get some kind of analog, the so-called biofuel, has been real for a long time. It will burn just as efficiently. In 2013, there was a study where bacteria were taught to eat sugar and convert it into hydrocarbons. The authors argued that it is quite realistic to replace sugar with manure and organic waste. As for me, very cool. For more recent research, even cooler. How about synthesizing fuel literally from the air? Well, more precisely from carbon dioxide. We have found a suitable bacterium that can take free electrons from the environment for its metabolic pathway to reconstitute carbon dioxide from the air into butanol and other hydrocarbons. It's green energy squared. And now I will turn everything around. If you think that everything is more or less fine with the ecology now, I will upset you. It's worse than ever now, but this is a topic for a separate video. However, classics, oil spill for example. Such tragedies will continue more than once. Another example is plastic that formed the Great Pacific Garbage Patch. Some call it garbage continent. So that you understand, there are only about 200 countries in the world, and if you establish a garbage country on the area of this spot, then it can enter the top 20 largest countries in the world. This spot is bigger than any European country. Something needs to be done with such things, and it already do. And as one of the options, use bacteria that eat plastic and other hydrocarbons, processing them into water and carbon dioxide. Such bacteria appeared as a result of evolution in places of plastic processing. They were found in 2016 in Japan. It turned out that these bacteria are able to destroy ordinary plastic in 6 weeks, when it usually takes 100 or 2 years. Why not take their genes and give them to more controlled or rapidly multiplying bacteria? Of course, there are many difficulties here, but the prospect is very interesting. Now let's move on. It was all just about bacteria. Now let's talk about plants. That's where people are most afraid of GMOs, focusing on the saying "you are what you eat." There are a lot of arguments here. Let's start with the classics. Therefore, I have no doubt that most of you know this joke. Here are 3 pictures for you. Put it on a pause and write what are these fruits and vegetables. How did you not find out? It's banana, eggplant and carrot. Just natural, without human intervention. The banana was barely chewed and had seeds. This natural carrot has been ripening for as much as 2 years. Extremely effective, isn't it? People have changed plants with the help of natural mutations and crossing the right mutants with each other. That is, people literally mixed up genes. And tell me, why is a smart gene change considered worse than a random one? Naturally, this is not the case. Moreover, when we use the methods of genetic modification, we know what we are changing. And if we just cross different types of plants, you can get very dangerous specimens. A classic example is the Lenape potato. I don't know how to pronounce Lenape, but Okay, it was obtained in 1967. People crossed two types, so that it turned out to be a very strong, large potato, which is ideal for chips. They began to grow and sell. But how did they know that there was a poison inside this randomly obtained variety? Inside, a couple of factors were randomly combined, which in total became a toxin. But the potatoes have already been released to the market, and people began to poison themselves en masse. Is this naturalness still so good? People are afraid of what they don't understand. Therefore, we just need to explain to them how it works. Now I was talking about the difference between GMOs and breeding, showing its disadvantages. And what are the advantages of GMOs? What are examples? From the obvious, this way you can increase the fruits, increase the resistance of plants to diseases so as not to treat with pesticides. It is also possible to reduce the amount of water necessary for growth, etc. But this is all uninteresting and obvious, because these are the main tasks of scientists. I'm interested in the fact that life is a very interconnected thing. Everything consists of the same substances. Therefore, what bacteria can do, plants can do the same, just with some special features. I have already talked about biofuels in bacteria. This can also be done by a plant called jatropha. This is a plant whose seeds consist of 40% of oil. This oil can be slightly changed and get almost diesel fuel. This was done around the 2010s. Analogs were also developed, for example from corn. The same can be done with cleaning up the earth's pollution. For example, at landfills on the battlefield a lot of things explode. TNT or hexogen, this kind of substance. And as a rule, not all explosives burn. It happens that the projectile does not explode, and after years the contents begin to leak out, poisoning drinking water and soil. At such landfills, the researchers found a bacterium that secretes a protein that decomposes these explosive compounds. We pass this gene to ordinary grass, and that's it. Simple grass decomposes explosives. Well, as with bacteria, plants can produce medicines in the same way, or something like that. The classic here is golden rice. I don't know about you, but I was told as a child, eat carrots so that you have a good eyesight. As it turned out, I was something lied to. It was more correct to say eat carrots so as not to go blind. Of course, it's about vitamin A or retinol. He's heavily involved in the work of vision, and without it, night blindness will develop first. Imagine that. The sun is setting, twilight, summer lights turn on, but you can't see anything. This is night blindness, when the eye cannot adapt to low light. But this is just for starters. Then you will have a so-called dry eye. That is, tears disappear. Not the ones you cry with, but the ones that leave your eye wet. Well, then the eye completely shrivels and the person goes blind. I would show you all these photos, but YouTube will ban me. So, why I'm telling you these horrors? You probably have not encountered this because you eat normally. But not everyone on the planet can eat a full and varied diet. And for example, in Southern Africa and South Asia, children regularly go blind and moreover die because of this. The fact is that blindness is only part of vitamin deficiency. Since the 2000s, 250 to 500,000 children have been sick every year, half of whom died a year after being blinded. So many people don't always die in wars, and here it's just kids. To solve this problem, you can produce vitamins and import them there en masse, or to import carrot-type products to these regions and distribute them for free. But it is clear that it is very expensive. But what if we take what the people of these regions eat? Rice. And to make this retinol or vitamin A, synthesize it in it. That's how the golden rice was created. It can save a bunch of kids. And then people will say that GMOs are bad? About plants, you can repeat the same thing that I told you about bacteria. We throw plants into a bioreactor and they produce interferon, erythropoietin, globulins and other proteins needed for medicine. But there is an even more fun thing. You know COVID totally sucks, and you know that vaccinations save lives. If you think differently, unfortunately, it only means that are you not aware. And by the way, I think it's not your fault. This is the fault of the government, which broadcasts very little educational content and does not develop the educational system. Although it depends on the country in which you live, and this is a separate topic. So, people are afraid of vaccinations. And what if immunity could be obtained without a needle? And after eating, for example, potatoes. After all, it's the same here. The complete genome of the coronavirus was known after how long? A week after the start of infections, I just don't remember. We take the protein from which the coronavirus spikes are made and force these harmless spikes to synthesize potatoes. The vaccine synthesizes itself. No laboratories are needed for the synthesis of the vaccine, its purification, etc. However, this is certainly our future. Edible vaccines are still only being developed and studied on animals, but there are already such edible vaccines for hepatitis B, C and E, measles, foot and mouth disease and cholera. Well, this is something about medicine. There is also a purely scientific application. The classic for which the Nobel Prize in Chemistry was awarded in 2008 is a green fluorescent protein. It was found in jellyfish in the 60s. This protein can absorb light in the blue and ultraviolet spectrum and emit back in the green spectrum. This protein has a whole history of change and research, but as a result, now everything is brought to mind and you can get a pretty bright, independent, almost any color of glow. Again, the question is, for what? For research. And that's how we smoothly move to the next step after plants, to animals. To improve or cure something, you need to know the mechanism. For example, how is the nervous system formed? Its development or work is quite difficult to observe, because it is a mass of overlapping cells that you can't tell apart from a living one. To do this, the cells are usually painted, but having already cut off a dead piece. So, we can attach this glowing protein to nerve cells. If you do this with the transparent fish, then you can track how the drug for anesthesia affects its development. But not all animals are transparent, and sometimes you need to look into the cells themselves. If they all glowed green, nothing would be discernible. But if we give them different shades available to us in random order, then each neuron will glow with its own unique color. This technique was called rainbow, well, like a brain and rainbow. To explain something here, I don't see the point. Everything is as clear as possible, very informative and even aesthetically pleasing. And at the beginning of the video, I said that you can do it tricky, so that proteins start to be released only under certain conditions. This can also be done for green fluorescent protein. There are really dozens and hundreds of variations. For example, you can make a protein that first glows green, but after a while, under certain conditions, it turns red or it just stops glowing. And if the conditions are normal, then nothing changes, a kind of indicator. It would be possible to give examples on the oxygen starvation of fruit flies, for example, but it's not that interesting. Let's start again with the fish. Around the 2000s, green fluorescent was implanted in DNA of fish. The idea was that when toxins appear in the water, the fish feel it and begin to glow. A very convenient indicator, isn't it? Besides natural and beautiful. It was pretty obvious, so the fish were registered under the trademark glowfish. And in theory, you can find constantly fluorescent fish almost any color. Isn't that cool? Exactly the same thing was done with mice, calling them neon mice. Here they are running. Pigs were also made green, and by different teams of scientists. There are even glowing green cats, and more than that, glowing monkeys. All this, of course, is not for nothing, but for different studies. For example, in cats, this method was used to investigate the transmission of FeIV, the feline analog of HIV. Simply put, transmit it, infect it, you will glow. And the story of monkeys was that scientists wanted to study diseases of the nervous system, such as Parkinson and ALS, which Stephen Hawking had. Well, again, illuminate and explore anything. The next story is no longer about pure genetics, but it is logical to mention it right now. This is optogenetics. Here we return to the topic of the eyes again. Maybe you remember from biology lessons that the retina is responsible for our vision. There are photosensitive cones and rods. So that's how it works. Again, thanks to the protein. There is a protein rhodopsin on the rod cells. If light hits this protein, then it excites the optic nerve as a result of complex reactions, and you get a signal. That is, you see. Speaking of which, this protein, rhodopsin, consists of two parts: slightly modified vitamin A, or retinal, and opsin. Therefore, without vitamin A, our eyes stop seeing in the dark. That's how everything is interconnected. And not only that, it's still more connected here. Life is not only built on the principle using DNA, but since ancient times it uses approximately the same proteins. Do we have rhodopsin? So it will be in cats, elephants, sharks, etc. It turned out that even one algae has a variation of it. Having studied its variation of rhodopsin, it turned out that in some sense it is simpler and it can be embedded in any nerve cell, not just in the eye. That is, a nerve cell with this protein will be excited if you just shine a light on it. This is the highest accuracy. Not a pill that affects the whole brain and not the electricity. You can excite individual brain cells. Speaking of which, to check that the nerve cells we need have received this light-sensitive protein, we can also throw a green fluorescent protein into them. If the cells glow green, then everything has succeeded. Well, now, showing how everything is interconnected and how optogenetic works, I will explain what scientists have already tested on mice. For example, they could affect motor neurons and control the movements of mouse whiskers. In some experiments, mice were forced to forget previous experiences. That is, they influenced the transfer from short-term memory to long-term memory. Well, with some tricks, they did the opposite, created false memories. Similarly, they affected the sense of smell, vision, and more complicated things like emotion, causing fear from scratch, or on behavior, making the individual more sociable or less sociable. That is, the most accurate ways of controlling the brain appear. Plus, you can also affect muscle tissue. They are also sensitive to excitation. So the researchers were able to regulate heart rate problems. And in another very specific case, proteins were attached to the sexual organ of males in mice. And now, when blue light is shining on them, there is an instantaneous excitation of this organ. Well, of course, this is a practical application, which is more interesting for me. In fact, there are more research tasks there, for example, how Parkinson's disease works, etc. But nevertheless, genetic engineering has long been not some new science, because there are such new related areas, highly specialized. Another narrowly focused topic is cloning. Only Dolly the sheep is almost 20 years old. Of course, cloning is very advanced, although it is still experiencing huge difficulties from an ethical point of view. But this is if we are talking about people, and then so far. But what about bananas?
Banana. Banana.
I have already shown wild bananas. They contain seeds, which then in theory should germinate. Well, you know how it works. And then, how do new trees with bananas grow? It's very simple. They are being cloned. Of course, it sounds much cooler than it actually looks. But the trick is that it's pretty easy to do with plants. You can take part of rhizome, transplant, and essentially the same organism will grow somewhere else. So, in about the 1850s, the British William Cavendish did. 100 years later, his bananas became the most popular and you're still eating them. That is, we have been eating fruits, in fact, of the same organism for 170 years. As I said, there are no problems with cloning at all. Dolly the sheep and bananas are very simple and familiar. Then how do you like cloning clones? This is also done. For example, the Japanese in 2013 cloned the same mouse 25 times in a row. It's really something like
200,000 units are ready, with 1 million more well on the way.
Clones in movies lose their temper when they find out that they are not the originals. What if he finds out that he's not even a copy of a copy? I think now we are ready to talk about genetically modified humans. Yes, a person has not yet been cloned, at least officially, but they did an equally interesting thing. They made up the genome not of 2 people as usual, but of 3. And not in the way you thought. The fact is that our genes are contained not only in the nuclei of the cells, but also in mitochondria, such plants for energy production. These mitochondria are inherited essentially on the maternal side. The mother is okay, but the baby can't be born because of breakdowns in the mitochondria. And then you can do in vitro fertilization, when an egg is fertilized in the laboratory. But just at this moment you can plant mitochondria from a third person. This is called mitochondrial replacement therapy. Thus, the child genome is essentially from 3 parents, and there are already about 40 such children, well, according to the data for 2016. And this is just the beginning. There are enough tricks with different parents. Scientists are now working on a very interesting question. Look, there are stem cells everywhere inside us. For example, in the red bone marrow, they are such an endless supplier of new blood cells, infinitely division and turn into erythrocytes, lymphocytes, etc. The same is true in the testicles and ovaries. In men and women initially there are generally the same cells. It's just that when the stem cells mature, they turn into a sperm and in others into eggs. And if we simplify the meaning of the process, then we can say this. Scientists are now on the way to creating both an egg and a sperm from your stem cells, at your choice. And then take any options. A woman can get pregnant from artificially created spermatozoa of another woman. One man can fertilize an artificially created egg from another man, although it will still have to be carried by a donor woman. And the most interesting option, a woman can make her own artificial spermatozoa and fertilize herself, having given birth to a very interesting person. Who do you think it is biologically? Clone or something else? Write in the comments. This will literally become real in practice within the next few years. Here is a very recent article that tells how the Japanese made spermatozoa from stem cells. There are still buts there, however, this is already a reality. By the way, let me remind you, our genome consists of 23 pairs of chromosomes. In theory, then will come a time when you can take a chromosome from any person and make a child out of 23 parents. And if you really try, you can make a child out of any genes. Take the eyes from this person, the eyebrows from this, the height from that, etc. But this is absolutely the future. I decided to start with cloning and birth because At this time, it is most convenient to modify a person. And as you already perfectly understand, there are no difficulties here. The most high profile, and in fact the only case related to genetically modified people, was in China in 2018. In short, He Jiankui provided in vitro fertilization services. Everything is quite legal, except that he decided to change the genes of two children. Their names are unknown. The idea was to turn off the protein that the HIV virus clings to. As a result, these children were born healthy and immune to HIV. As it turned out later, this protein has already been experimented with, on animals of course, and when this protein was removed, the subject's memory and recovery after strokes improved. So the children may be even smarter than could be. The scientist was given 3 years in prison. This year he was just released. So, genetically modified children are quite real today. People are limited only by laws and opponents of this direction. But everything we talk about today is absolutely possible. It is realistic to make sure that people are born completely healthy without any abnormalities or disease. Really make a glowing person. It is really possible to make sure that when you are shown with ultraviolet light, your mood rises or your hand suddenly rises. If you want a person to be pumped up and not store fat, while sitting in a chair 24 hours a day, this is also real. There is such a breed, the Belgian Blue. Their thing is that they look like characters from JoJo. Why are they so pumped up? Because of random mutation. They have problems with myostatin, a protein that suppresses muscle growth. Accordingly, their muscles grow just like that, and even almost no fat is stored. In general, I probably wouldn't mind this. All this very real for an unborn person. But what about you and me? Viruses quite effectively introduce their DNA into our cells, which is why we get sick. So there are no problems. We plant in the virus what we need to synthesize and let it into human cells. As a matter of fact, this is how regular vaccinations against the same coronavirus work. We take the adenovirus, insert into it only part of the instructions for the synthesis of COVID, that is, only the crown spikes. They begin to reproduce in us, the immune system sees someone else's protein and begins to develop immunity against it. In the same way, only with more advanced methods, gene therapy works. In practice, it started working from the 90s. The first such treatment was given to a girl with a mutation, because of which one protein was not produced, leading to immunodeficiency. She had to walk all her life in a bubble and still at some point get infected and die. The effect of such vaccination was temporary and people needed to receive new doses. But in the end, the girl is still alive and fully active. After 30 years, gene therapy is developing and becoming more accurate and long-term, so that you don't have to constantly inject a new dose. The main directions, which is logical, are the fight against genetic diseases, viral diseases and with cancer. Because cancer is essentially a genetic breakdown. Patients receiving such treatment are in fact GMO people. And actually all that stops scientists from changing healthy genes, not sick ones, are laws and ethics. The thing is, as you can see, nothing I've told you is a few years old. Everything I was telling started with this was invented in 80s, this in the 90s and this in 2000s. The problem is that around these 2000s, various kinds of problems occurred. There were escapes of such genetically modified crops into the wild, which caused scandals. Allegedly, now these mutant plants grown in laboratories will destroy our nature. When using gene therapy at the very beginning, there were a couple of deaths and everything like that. This greatly concerned the government and eco-activists. Therefore, around the end of the 2000s, the beginning of the 2080s, the laws on the use of everything genetically modified were greatly tightened. Plus, for a reason at the beginning, I gave statistics. People don't know what GMOs are. For example, when laws were introduced obliging manufacturers to label a product that it contains GMOs, activists also suggested adding a sticker stating that the product as a whole contains genes or DNA. I wonder which products would not have this label. In salt, perhaps? Or the golden rice I told you about. There were some locals against that. There was a case when they uprooted a whole crop of such rice. And the famous Greenpeace in 2005 opposed golden rice, fearing the spreading of GMOs. And we haven't even talked about religion, which says that genetic engineers are playing God. This video I want to show how everything is interconnected here, and that GMOs are just conscious use of the mechanisms that we already use. It's better to consciously add something to the plant than to hope for a random crossing in attempt to get more lush fruits. As it was with the Lenape. We are all genetically modified anyway, because we were vaccinated, and even if they didn't, then a dozen other times they were sick with viral diseases that terribly randomly change our DNA. Therefore, you will not find a single article from serious scientific publications that would talk about the harm of GMOs. Neither about bacteria, nor about plants, nor about animals, including humans. I hope the younger generation is watching me and it will grow up without this prejudice and fears, more educated, and some of you will find, for example, gene therapy for aging, also a genetically programmed thing. It would be nice. What do you think? Write what you think about all this. And if you were interested, share the video with your friends and press the like button. Let me remind you that this was my first video. I hope you liked it. Write what can be improved. I will try to do it next time. So subscribe. See you soon.